Chapter 5, Section 2 is over mitosis and cytokinesis. Cells divide during mitosis and cytokinesis. The mitotic phase, or the M phase of the cell cycle, includes both mitosis and cytokinesis, and it alternates with the much longer interphase. Interphase itself will account for about 90% of the cell cycle. Chromosomes condense at the beginning of the mitosis phase. We start with DNA, and that DNA slowly starts coiling around these proteins that are called histones. Think about thread wrapping around a spool. It helps keep the thread organized, and it allows you to pack a whole lot more in a small area. Same concept, but instead of having a spool for our thread, the spool is this histone protein, and the thread would be like the DNA. Once it starts coiling up on itself, much like an old-fashioned phone cord, I don't know if you guys remember seeing those, but they're very, very coily. They keep curling and curling and curling and spiraling until it gets so tight it starts coiling up on itself, and it becomes this super coiled DNA. And once it's so coiled up, it forms this chromosome structure we see here. So this DNA will wrap around the proteins, or the histones, that condense it and cause it to take up a much smaller amount of area. So DNA plus proteins is simply called chromatin. We'll talk about chromatin when we start looking at images of cells that are dividing. 1/2 of a duplicated chromosome is a chromatid. So this would be my original chromosome, and after it duplicates itself, it makes an identical copy of itself. Now I have 2 chromatids. We call those sister chromatids. They are identical in every way. The sister chromatids are held together in the center point at what we call the centromere. It's almost like if you had a little Velcro dot holding it together. There's also some chemicals that hold them together throughout the length of the entire chromatid. We're not going to get too far into that. At the very ends, we find what are called telomeres. A telomere is kind of like an eraser on the end of a pencil. It's still important and it serves a function, and its job is to protect the DNA. So if you think all the good parts of the DNA are up here, the parts that code for all the proteins that make up who you are. This end right here, its purpose is not to control any protein production or genes. There's no genetic material stored here. It serves as kind of a buffer for this chromatid or this chromosome later. And what happens is every time the cell divides, you lose a little bit of telomere. So kind of like every time you use an eraser, you use a little bit of it until eventually it's gone. And once it's gone, essentially the pencil is useless. Chromosomes are made up of DNA that's tightly coiled around those proteins or histones, and that's different than a chromatid. So chromosomes are what we are generally used to seeing. They look like an X, but in reality they're not really an X. When we look at that picture of an X, what we're looking at is the 2 sister chromatids that are joined together. And those are just duplicated chromosomes together. The centromere is that center point of the chromosome. And then we're going to start talking a little bit more about genetics. And when we do that, we have to understand there is a diploid number for every species and a haploid number for every species. The diploid number, prefix di meaning 2, is just the number of chromosomes in a regular cell, that 2n number. So for example, human beings, we have 23 chromosomes, and they come in pairs, so our diploid number is 46. Our haploid number, which is the number of chromosomes in a gamete, or a sex cell, a sperm or an egg, is 1n. So our haploid number is 23. Like we said, a gamete is just a sex cell. It's either a sperm or an egg. And sex chromosomes. We're going to start looking at these chromosomes themselves and what kind of information is stored on them. Of those 23 chromosome pairs that you have, one of those pairs determines your sex, your gender. You are either XX or XY. Now in biology and science, there's always going to be those loopholes, those but what ifs. There are genetic abnormalities that occur. There's triple X syndromes, there's XXYs, there's things like that that happen, but they're very, very rare. All of your other chromosomes, the other 22 pair, or 44, are called autosomes. So when we look at the cell cycle itself, we have interphase, which is what the cell's spending 90% of its lifetime in. And then we have this mitosis or meiosis phase, depending on what type of cell it is. And finally, the cytokinesis, which is a very, very short fraction of a time. In mitosis, which is what we're going to study this chapter, we're looking at a regular cell, so like mito goes through mitosis. The cell is going to copy itself and then split in half, and it's going to result in two identical cells. During meiosis, which is next chapter, we're going to look at certain types of cells, sex cells, that will copy themselves, they'll split for the first time, and then they turn around and they split again. And that's going to give us 4 mismatched cells. And the significance is going to be when we start talking about genetics and how we inherit traits from our parents. So interphase, mitosis, and cytokinesis make up the parts of the cell cycle. During interphase, the cell is focused on growing and duplicating things. So interphase, think about like intermission. If you go to the movies, you go to a play, during intermission, what do? You get up, you walk around, you stretch, maybe you go get something to eat, maybe you go to the bathroom. But during that time, you're just kind of hanging out, growing, doing everyday cell things. This is the phase that the cells are going to spend their time growing, copying the DNA, and then preparing for cell division. We call those G1, S, and G2 phases. The DNA that's located inside the nucleus, we can see, is in the form of chromatin here. So if we were to look at a cell in reality that was dividing, you wouldn't be able to see chromosomes. You wouldn't be able to see those thick rope-like structures. You wouldn't see the X's. You wouldn't see any of that. You would just see kind of a hazy gray area, and more importantly, that nucleolus, which is just that dark condensed area of DNA inside the nucleus, and its job is to make ribosomes. So mitosis divides the cell's nucleus in 4 phases. During prophase, chromosomes condense and spindle fibers will form. The DNA will start to shorten and coil up into rod-shaped chromosomes. It forms chromatids, then 2 copies of the chromosomes. So we see those chromosomes pairing up. That's where we get that X shape and then those homologous pairs, which is more like your right sock, left sock. Another thing to mention, prophase means pairs, so chromosomes will pair up. During metaphase, they're going to line up in the middle. The spindle fibers that came from the centrioles are going to work to direct the movement of the chromosomes inside the cell. So you can see those spindle fibers here. They're colored in green. In here, they just look like these blue lines. Okay? And their job is just to line up those chromosomes in the middle of the dividing cell, much like football players on a football field. But keep in mind that we're looking at a 3-dimensional object here. And so it's actually they're lined up on a plane. So they could actually be next to each other. But if you tilt it a little bit, it looks like they're all lined up. So it can be a little confusing when you look at a 2-dimensional picture. But that's what we have going on. So metaphase, middle. They're all lined up in the middle. Now there's some other fibers, these non-kinetochore fibers, that are coming out of our centrioles, and their job is to simply grow and stretch the cell to make room for this next step. Our next step is anaphase. During anaphase, sister chromatids will separate at the centromere and slowly move towards opposite sides of the cell. And we can tell when we look at a picture that this is anaphase because you see the little tails of the chromosome, they're being pulled towards the outside. So anaphase means apart. And then finally we have telophase, the final stage of mitosis. During telophase, we have 2 cells becoming apparent. Chromatids reach the opposite ends. They start to uncoil as the new nuclear membrane forms. We see that cleavage forming. because remember, cleavage means crack in science, no matter what science you're in. In animal cells, we see this cleavage furrow. In plant cells, we are going to start seeing a cell plate form here as well, which is just vacuoles that are filled with the cellulose fibers that make up a cell wall. And they just start lining up in the middle and fusing together, and they form a little makeshift wall, and then eventually they close off, and then we have two complete cells. So we see them pinching in here, almost like if you were dividing bread dough. And then finally we have cytokinesis. Cytokinesis is going to look a little different in the animal and plant cells. So the animal cells, that membrane pinches closed, and that's it. Plant cells, the cell plate will form, and then finally a cell wall. The final stage here, we have two identical cells. They've both got their very own nuclear membrane. They both have identical copies of everything inside of each other. So they're completely identical. The actual footage of cell division happens very quick. So once those two new cells are formed, and we have that pinching off, this cell is now automatically, it's back into interphase, and it's going to spend 90% of its life in interphase. So cytokinesis is important because this is when the cytoplasm itself divides. So this is the final step. Cell division is controlled by checkpoints. If things aren't progressing correctly, then that cell is going to divert into the GO phase. The GO phase is the exit of the cell cycle. So at a certain point, if the cell is not growing and progressing and doing what it's supposed to be doing, it gets kicked out of the cell cycle. So it's not just going to keep passing on bad parts or bad genes. For example, my brain cells, my nervous system is in the GO phase. My nervous system quit growing and dividing when I was roughly about 21. And that's one of the reasons that 21 is that magical age where your nervous system quits growing and dividing at that point. And so if you were to suffer some traumatic injury, you're less likely to repair yourself and heal from it. And then, of course, we have our funny. Can you explain cell division very fast? You have your cell. It grows and elongates, splits into two, forms complete two. Ha ha ha.